Okay, we are live now. Hello and Assalamu alaikum everyone. My name is Arsalan Riaz, the Vice President of RSG Pakistan. RSG Pakistan is an affiliate of International Society of Computational Biology, which aims to educate and raise awareness among the Pakistani community about the latest trends in computational biology and bioinformatics. I welcome you all to the another exciting episode of Computational Biology webinar series. Today, our keynote speaker is Dr. Safee Ullah Chaudhary, who is a Ph.D. at LUMS, where he established biomedical informatics research laboratory, which is now involved in the development of cutting-edge techniques in proteogenomics, system biology, and e-health towards the scientific wellness. So, thank you, Dr. Safee, for joining us and for sharing your knowledge with us regarding the model-driven personalized oncology. On to you, Dr. Safee.
Thank you very much. Assalamu alaikum, everyone. So, the topic of my presentation today is about a project that we undertook during the last 3 years. And the project was about developing a personalized therapeutics platform uh, and a pipeline for treating cancer patients and devising optimal chemo and targeted therapy combinations for their treatment. So for that, we employed an in silico Drosophila patient model, and we achieved various novel combinations for treating colorectal cancer. But to before I get into the details, I'll just give you an overview of what exactly cancer is and its complexity. So the disease begins with dysregulation in various molecular pathways and then escalates up to tissue and organ level. The spatiotemporal diversity within these aberrations, it gives rise to plasticity and vast phenotypic heterogeneity. And this heterogeneity is one of the biggest impediments in treating cancer. So such diversity at the genomic as well as proteomic scales and their heterogeneous manifestations at the cell and tissue level were summarized by Hanahan and Weinberg in one of their landmark review papers titled Hallmarks of Cancer. And of course, then they also did a version 2 of that, the next generation of Hallmarks of Cancer in 2011. So, briefly, to summarize the hallmarks of cancer, you can see this Circos chart, wherein you have various properties of cancer, such as sustained proliferative signaling, evasion of apoptosis, and independence from growth factors. So, all of these can come about through a variety of mutations in various signaling pathways. Now, individual mutations can impart a single hallmark, but the crosstalk between various mutations and the hallmarks results in the heterogeneity that I was talking about earlier. As a result, uh, patients, when they go to cancer hospitals, they end up presenting deep plasticity to the clinicians. And the symptoms, they get treated with the same medicine as a result, but in some cases, the efficacy is wonderful. In other cases, there is no effect on the patient. And yet, in some other cases, there might be some side effects which results in perhaps the death of the patient as well. So this is the current state of the art in treating cancers. However, sorry, so we propose a futuristic personalized diagnostics and treatment pipeline in which every patient can go to the hospital and have his or her cancer treated in a personalized manner, wherein the mutations that are within that specific patient are considered and factored into the therapeutic regimen. As a result, the efficacy can be enhanced and everybody can get a treatment that works for them. So, to give you an example, so in case of imatinib, which is a treatment of choice for CML patients, we have seen a lot of resistance against the drug, and more so, there are toxicity issues with the drug. So, a couple of interesting cases that need to be brought for such a case is that of Lucas Wortman. And so it's a very interesting case. So he works in Washington University's Cancer Center, and he had lymphoma. So what happened was that he got treated from a drug which could not pass the FDA approval, and he got well. So it is not essential if there is a drug and it doesn't work for a specific cohort of patients, it will not work for anyone. So we can repurpose that drug, we can reuse that drug in light of the patient mutations. Similarly, another case in front of us is that of Dr. J. Martin, and he also got healed from a drug that failed trial. So there are several such stories wherein cancer patients, they survived as a result of making attempts using personalized mutations and selecting therapies in light of those mutations. So the role of next generation sequencing comes in at this point. So now we know that this is the age of high throughput genome sequencing. Of course, it all started in 1977 with Sanger sequencing, and then PCR was developed. Human Genome Project got completed in 2003. And now people have started accumulating genomic information. So, not just that, but also since the high throughput nature of sequencing has come about, you can simply go to the cancer hospital and get yourself sequenced, and the treatment can be therefore selected in light of your mutations. However, there are several challenges using these multi-omics studies. So firstly, the sample preparation, study design, and reproducibility, as is the case with most of the studies. Also, you have, individualized omic data sets. You need to normalize them, and you need to evaluate how to compare various data sets with each other. And then, of course, there are false positives and p-value issues that you want to really evaluate before declaring a sample as a normal or a cancer. So once you have done all of that, you need to stitch all of this into a workflow such that this workflow or pipeline is available to the oncologist in the hospital. And you just go in, you input your sequence, and you get efficacious therapeutic combinations as an output. So but once you're able to do that, then comes in that you need to evaluate these combinations, and we don't have a lot of whole animal models for that. So in the lab, one of the most famous models that we have is the Drosophila melanogaster, or the white fly. It's a small fly that you can see on bananas these days. So you can reproduce the model real quickly, and the life is short, and you can conduct any kind of genomic editing and create a copy of the human mutational status. There are other models as well, such as the mice or the chimp model, but of course the time to rear them is long, so it's difficult to use them in a high throughput fashion. So in terms of Drosophila and its use as a whole animal model, uh, we will have to, and in context of colorectal cancer, we'll have to see what is the structure of the colon in the Drosophila melanogaster. So if you look at the entire length of the gut, then you will see that there are various segments: the foregut, the anterior midgut, the middle gut, posterior, and the hindgut. So in our case, we have selected Drosophila melanogaster and its posterior midgut, as shown here, to create an in silico version of the fly. And we have looked at the posterior midgut and observed what kind of cells they line the interior lining of the posterior midgut. So there are 5 types of cells that exist in there. These include the intestinal stem cells, enteroblasts, enterocytes, enteroendocrine, and visceral muscle cells. Now, once we set out at creating an in silico fly, or at least its midgut part, literature review showed us that such a patient model already existed. So Bangi et al. Have produced such an in vivo model wherein they have a fly, and they have introduced patient specific mutations, KRAS mutations in particular for colorectal cancer, and created a fly version of colorectal patients. So this was a lot of encouragement for us, and they used this platform to identify trametinib plus zoldronate as a KRAS mutant metastatic colorectal cancer treatment. So summarily, patients with treatment resistance to KRAS in colorectal cancer could be evaluated by using their FLY model. And they screened 9 key mutations, and they were able to identify the combination of trametinib and zoldronate as a candidate treatment. So this was published in 2019 by Bangi et al. So what we then did was we took up the same idea, wherein we went one step further. So instead of having an in vivo fly model wherein you have various mutants and you have to derive the transgenic models, we created an in silico version or a computer version of the Drosophila midgut. So for that, we took up the midgut version, as I was alluding to earlier. We looked at the variety of cells that were available there, and we created their biomolecular networks. So the biomolecular network or regulatory network is the governing structure for cell fate programming. So if you want to see how cell fates are programmed in the midgut of the Drosophila melanogaster, then you can simply create the entire pathways in silico and look at their crosstalk and their cell fate programmability. Therefore, you can bypass the creation of in vivo models. So the pipeline for converting the in vivo Drosophila patient model into an in silico counterpart involved 5 cells that were taken from the midgut section. We created the biomolecular regulatory networks for each one of these 5 cells. We looked at the robustness under 10% perturbation, and then we put the networks under normal, stress, and cancerous conditions. So a normal cell under normal conditions, a normal cell under stress conditions, a normal cell under cancerous inputs. So then we saw the behavior of these networks and we validated it from the literature. In the next step, what we did was we looked at the gut sequence database, which is an RNA-seq database for expression database for the midgut. So by looking at the expression of various genes that were reported in the flygut-seq database, we came back to our model and validated the correctness of our model. So we validated the model through robustness, through evaluating the network sensitivity to stress and cancerous inputs, and thirdly, by looking at the RNA expression given in the flygut-seq database. So once we had established the model, in the second row here, you will see, so we took this model and we induced or introduced colorectal mutations that were reported by Martorell et al. In CRC. So there is a progressive sequence of mutations that occurs in colorectal cancer, and we use the Martorell study to input such mutations into our network model. In the next step, we evaluated 9 FDA approved drugs on that network which carried the mutations of CRC. And as a result, once we had the network going with Martorell and the 9 FDA approved drugs, we input patient expressions to personalize the networks to specific patients from the TCGA database, or you can also access it through GDC or cBioPro. So once we had done that, we saw how patients responded to various therapies. So this is the story of how we developed an in silico Drosophila patient model, and we are able to screen. We were able to screen 10 patients for a study that we just published in Frontiers of Oncology in a special issue on combinatorial therapeutics. So once we have done that, you have treatment for specific patients, but the story doesn't end there. Next up, what we did was we created combinations of all the nodes in the network, and we performed an exhaustive screen to evaluate which target might be the most efficacious, irrespective of whether an FDA approved drug existed or not, because there could be targets that can be developed further later on for drugs. So, and then once we found such targets, we looked at the pan drugs database to see if any one of those targets was actually given there. And then the targets that were there in pan drugs, we used them to create novel combinations of targeted therapies along with chemotherapy for CRC patients. So this is the overview of the story that I'm going to discuss today. So first of all, we built, as I indicated, we built 5 biomolecular network models. So this is just one of them for intestinal stem cell. And we created logical rules to represent the regulation in this network. And as you can see, here is an example for you. So in case of Jack, it is regulated by SOX36E and DOME. So the rule, the logical rule for this is stated as Jack equals not SOX E and DOME. So this is how we listed down all node regulations. So we did this for 5 different cell types, including the intestinal stem cell, and we created a screen that helped us to evaluate the robustness of the resulting networks, as I was talking about earlier. So the cell-fate programming that emerged from these networks is shown here. So as you can see, the intestinal stem cells, they had a lot of apoptosis and extrusion under 10% perturbation, which is still considered normal. And the enteroblasts, they had a lot of apoptosis and extrusion as well. The endocrine cells, they had apoptosis and DPP production, which is a hallmark in case of a normal uh, fly midgut epithelium. The visceral muscle cells are shown here at the end. So once we had evaluated the robustness and perturbation of the network, we established that the networks we created follow power law or Pareto's network. So all networks, all biological networks should be robust if they are to regulate biological function. So having established robustness, we moved on to evaluate the self-hit programming capability of these networks under a variety of conditions. Here you can see blue, orange, and gray in the bar chart given below. So blue is for the normal condition, the orange is for stress, and gray is for cancer. So as you can see, the gray bar in intestinal stem cell for apical and basal region, as well as for enteroblasts and enterocytes, showed a lot of multi-layering. So this is how a normal cell behaves under cancer conditions. So this is also validated. So now we have a network. We have 5 networks that are robust and that are showing good response in stress and cancer, as reported in the literature. So we went to the RNA-seq database, the flygut-seq, and we compared the outputs of our model with the experimental data that was reported there for various output nodes. So the output nodes, as shown here in the 3 panels A, B, and C, tallied really well with the experimental data, which is shown in blue, while the outputs from the model are given in gray. So as you can see, there is a very nice concomitance between the experimental and model results. So moving on, in the first case study, we replicated colorectal tumorigenesis under progressive mutations using Martorell's published CRC model. So the progressive mutations included APC, RAS, and APC plus RAS, that is together. So we then compared the output from our networks with Martorell's study, which was published a good 6 years ago. And we could see a very nice comparison as shown here in the at the bottom. So we have sulfates for various cells. So apoptosis, proliferation, differentiation, which is only in case of EB, loss of polarity, multilayering, and extrusion. So you can see as the mutations they increase, or for that matter, in case of APC plus RAS, you have apoptosis going down and proliferation going up. So this is a hallmark of cancer, as well as multi-layering, which is here in the bar chart as well. So after reproducing Martorell study and validating the trends, we moved on to see the gene expression confirmation. So as you can see here, we took up the data from the gene expression database and we compared it with the results that we had. So in case of control, so the APC arm was 0.698, while in case of APC plus RAS, we had a very lowered expression of APC arm. So here we can see that is shown here. So all of the genes that were upregulated and downregulated have been tallied. So this is important that once you create a model, it is validated against published studies. In the second case study, we employed Markstein's model to perform therapeutic interventions to validate the cytotoxicity of 9 FDA approved drugs. So Markstein reported that 13 FDA approved drugs had the potential of targeting colorectal tumors. So they distributed these into class 1 and class 2 drugs, wherein class 1 drugs do not have any adverse effect on wild type cells, although they do affect the tumor intestinal stem cells by reversing cancer. For class 2 drugs, the effect on the wild type is increased, as well as they promote proliferation in them. So it works against cancer cells. So Markstein et al. Claims that due to the microenvironment that is present in the midgut, the wild type cells are also getting affected by the drugs. Therefore, it is important to take into account the microenvironment of the cells. So, anyway, we evaluated proliferation in class 1 drugs and we evaluated proliferation in class 2 drugs. So the results tallied really well. So again, validating our robust network and its self-hit programming capability. So apoptosis in class 1 drugs is shown here in panel C, and apoptosis in class 2 drugs is shown here in panel D, so which is consistent with the results we obtained from our study. So we moved on back to Bangi. So Bangi had created flies with these, mutations for treating KRAS mutant CRC patients. So what we started doing was to create an in silico counterpart of such a fly, in particular in its midgut. So we ended up reproducing Bangi's KRAS mutant CRC model for evaluating optimal personalized treatment by incorporating key patient specific mutations into the model, and we also screened it afterwards. So this was a very nice case study that evaluated and gave us, the combinatorial therapies that were effective in the in vivo model and the in silico model. So the results as shown here, in case of trametinib, you can see that apoptosis is increased as a result of treatment. In case of mutation, of course, you see in the middle of panel A, you have apoptosis and proliferation with an equal propensity. So if you incorporated zoldronate, the treatment resulted in a reduced amount of apoptosis, while proliferation was still observable in the treatment. So if you created a combination of trametinib and zoldronate, you can see that trametinib and zoldronate combination completely wiped out proliferation while apoptosis was enhanced. So we propose that the combinatorial therapy using in silico Drosophila patient model, which involve trametinib, zoldronate, and trametinib plus zoldronate, can give you a variety of results in the light of your patient expression. So this was the output of the in silico replica of Bangi et al.'s work. Next, we move to CBio portal, which serves you with patient-specific data, and you can from TCGA, and you can also access this data through Genomics Data Commons. So we looked at several patients, and we took up their mutations. So here in the green column, you can see uh, the nodes that exist in the network, while in the white column you have the mutations that were there in patient, and in the top row you have patient IDs. So patients that had a lot of mutations due to the heterogeneity and the passenger mutations, we summarized them through nodes that were present in our network, and then we treated these patients in light of their mutations using combinatorial therapies for each one of them. So the results from the identification of network nodes in cancerous conditions for exhaustive screening are shown here. So what we do is we take the entire network and we take every node and its combination with other nodes to create self-fate programming profiles, and you can actually see which profile is most beneficial for the patient, thereby reverse engineering those targets into and translating them into therapies. So we surveyed the existing literature and listed down the oncogenes and tumor suppressors which were present in our network. So we used this data to regulate, upregulate or downregulate the nodes in case of tumor suppressor or oncogenes for therapy. So they are both given here, and as you can see in this slide, so who identified efficacious node targets from the ISC network against patient-specific mutations. So in the first column, you can see the patients, and for each patient, you have proliferation and apoptosis as two cell fates that we are looking at carefully during the in silico analysis of the network. So proliferation is supposed to go down while apoptosis is supposed to go up. So this is how we evaluate the efficacy of various combinations. So oncogenic fate propensities were observed and the combinations were created using the nodes in the network. So once you have the nodes in the network, then you can look at pan-drugs database, and the pan-drugs database can help you find which drugs are available that act on those nodes that you have shortlisted, thereby enabling you to treat the network. So the potential drugs taken from pen drugs in our case were uh, regorafenib and bortezomib and imatinib. So of course, paclitaxel is a chemotherapeutic agent. So we create combinations of not just the targeted therapies, but also chemotherapeutic agent along with some targeted therapy, such that we have the highest efficacy in favor of the patient. So finally, the personalized therapy for each patient and its literature validation helped us to compare the combinations that we report from our study and the combinations that are reported in the literature. So interestingly, the combinations that we reported from our in silico model were actually reported uh, in and undergoing clinical trials for a variety of cancers that are shown here in the table. So we validated the combinations, and finally we came up by selecting 10 different patients and extracting or eliciting nodes that were most efficacious for treating those patients. And then we created combinations of these drugs, which included targeted as well as chemotherapies, to see how best we can fight cancer back for each one of these patients individually. And this is the result that we got. So here the bars in blue are apoptosis, which is actually good, and it should go up as a result of treatment, while proliferation or red should go down as a result of treatment. So for various patients, we could see very nice results. For instance, in case of the first patient, we had a lot of apoptosis, 01C001, while proliferation was controlled as a result of bortezomib and paclitaxel co-treatment. While for some other patients, we couldn't get the perfect result, as is the case for patient 32904, which actually has a lot of that it can be discussed to lengths because the networks that we produce are not um exhaustive. So they are limited in the number of nodes. So they probably do not abstract or model the entire picture for all patients. So we'll have to continuously expand these networks such that they become a representation of the majority of patients. So this is all from my side. So this work was led by Mahnoor, who is now leaving for Michigan Ann Arbor for her PhD, and the software developers and the case study team um, is here. Lots of work done by a lot of people. So I would like to thank you for the opportunity to present this paper. And if there are any questions, I'll be happy to take them. Ji.
Thank you so much, Dr. Safee, for an incredible talk. Um, it might be very useful for the audience as well. There are some questions that audience want to ask. So
Yes.
Laiba Shah is asking, can we use synthetic models for such study as well?
Synthetic models of you mean gene regulation?
Um, I think so.
Yeah, so the thing is you can have CRISPR knock-ins and knockouts as well. So these networks, you can create them in a way that you have certain genes that are hypothetical, and you can have the output calculated from these networks. But once you want to create a control case or the wild type case, you need to knock out those nodes before you do that. So this can be done, but one has to really be careful with designing the scheme of how such a screen is run.
So Momal Agha is asking, do these drugs involve synthetic lethal strategies?
Sure, yes.
Okay. Annie Danish is asking, how were you able to identify the key significant pathways required for such models in the different cell types of you mentioned?
Yes, so any the thing is that the the cells that we had selected were intestinal stem cells, the enteroblasts, the enterocytes, the visceral muscle, and enteroblasts. So there were these five cells. Of course, ISC had two niches, so the apical and the basal niche. So we had to, create two versions of that network conditions. So once we look at each cell, we have to look at the biological function that cell does in the midgut. So if it's an ISC, the purpose is to proliferate and differentiate. So if there is an ISC that differentiates into EB, then that is one cell fate that it programs. If it is to proliferate and divide into two, so that is another pathway that is involved. So you have to actually see cell by cell what kind of biological function that particular cell is performing in the system. And once you're able to identify the function of that cell, then you have to, factor and decode that function into signaling pathways. So each pathway can perform a part of that function. So once you have all the pathways with you, you can integrate those pathways into a network. And once the network is there, then it kind of abstracts the overall picture. I hope I answered your question. G, Arslan, I think your mic is off.
Sorry.
Yeah.
So as a young scientist, uh, let's say I want to conduct such a study that you have presented, what kind of tools I need to learn and practice?
Hanji, so Arsan, there are a lot of tools that are available online, such as BoolNet. So it can help you analyze networks. Of course, there's a platform that we have published in Nature Scientific Reports about 3 or 4 years ago. So its name was Atlantis. You can use that as well. One of my students, Osama, so he published that. And now we have made a bioarchive of next generation platform Tyson. So it is public, and anybody can use it. It's for free, and you don't have to ask us for any kind of permission or whatever. And the manuals are there as well. So there are a lot of tools that are available. In our case, because half of my lab focuses on software development, so we develop our own tools in C sharp and MATLAB, depending on the need. So once we have the tools, then we perform case studies. So this Drosophila paper is actually a case study for the Tyson project, and there are several case studies in the works, which include modeling of neuroblastoma and breast cancer and oral squamous cell carcinoma in your oral cavity cancer, as well as the Warburg Effect and autophagy. So we do case studies and we develop the software. So together we publish them after that.
Yeah, thank you so much. Very interesting to see how you develop all these tools.
Yeah,
Okay,
thank you.
I think we don't have any more questions,
Okay.
and we okay, we have a question there. Did you consider other models that can be more viable than Drosophila?
हाँ जी बेटे, इसमें ये है कि ड्रोसोफिला शेयर 75% of its genome with humans. So we kind of prefer a model that is a good compromise between application and reproducibility. So Drosophila has a very short life cycle, so every 2 weeks you can renew it, and that it kind of represents to a good degree the cells and the tumors within the humans. So of course you can go for chimp as well. It has a higher homology, but then the thing is, to maintain the chimps in the animal house, that is a problem in itself.
Okay, thank you so much. I think now we don't have any more questions.
All right.
Yeah.
You're most welcome, Arslan. Thank you for giving me the opportunity to share a bit of our work. And if there are any details or questions that are still out there in the minds of kids, I'll be happy to take emails. And of course, the time is so short, I'm unable to explain a lot of things that go behind the scenes. So if there is something that is unclear, just shoot an email to me. I'll be happy to answer. I answer all the emails.
Thank you so much. Thank you so much for your time. And RSG Pakistan will be organizing such events, such are webinars for their audience, for the young scientists, to get a bit of computational biology, how computational biology work in the research labs. Thank you
Yeah.
so much. Thank you so much for your
Yes.
time. Uh,
Yes, Arslan, before I go, I would want to indicate to the audience that this manuscript might come online in the next week in Frontiers of Oncology. So if anyone is interested, you can tune in for that.
yeah, and a preprint is also available, I think so.
Yes, yes, correct.
So we will be ending this talk with a positive note that we will be organizing such events more in the future as well. Thank you so
Thank
much,
you.
and we will be ending this broadcast.